Hello, my name is Neil Spooner. I'm the chair of the Patient-Centric Sampling Interest Group, a not-for-profit organisation dedicated to educating on these novel technologies for getting biological samples from human beings. Really grateful to ASLM for inviting us today to take part in their ECHO webinars. We're very excited about this, and we're looking forward to presenting to you, but also hearing your questions and getting your feedback. We have planned a second event for the 7th of May, where we'll be following up with some more in-depth discussions about how these technologies might be useful for Africa and how we can help move forwards. So I'd just like to move on to our speakers today. Our first speaker will be Joleen White from BioData Solutions in the USA, who will introduce us to the existing patient-centric sampling technologies. That will be followed by Chris Hendriksz of a rare cause in the UK, but he also is based at Northwest University in South Africa. And the title of his talk will be When Infrastructure Fails, Why Dry Blood Spot are Africa's Lifeline for Biological Sampling. But with that, I'm going to hand over to Joleen and see you on the other side for the questions. Are you there, Joleen?
Yes, I am pulling my slides up. Thank you. Hi, my name is Joleen White, and I'm very honored to be speaking with you today. I do have a quick disclosure. There we go. Feel free to reach out to me on LinkedIn or wherever you feel most comfortable. I'm always happy to work with my African colleagues. I do have a potential conflict of interest to disclose, which is that I used to work for Gates Medical Research Institute, which is another grantee of the Gates Foundation. But since they also do issue some grants to some of these device developers and manufacturers, I did want to disclose that I'm not favoring one over the other. I'm really just trying to give you a broader view of what is out there that you may or may not have been aware of. I am using a lot of images under Creative Commons licenses, and the others are from product websites where I've tried to disclose the trademark owner where within the images and slides. So what is patient-centric sampling in general? The key item is that traditional, or what we're calling conventional blood sampling, is really just venipuncture, which means that an individual needs to come into the site, that they have a needle puncturing their vein, which they can see. And then there are vacuotainers, which rely upon a vacuum in the tube to pull the blood from the patient quickly into the tube. But this isn't necessarily the easiest route for the patient. So when we talk about patient-centric sampling, what we're talking about is a variety of factors. One is making sure that the right sample is collected, that you're not collecting samples that aren't needed. Using processes that are convenient for the patient, that aren't asking the patient to conform to your process, but rather developing a process that meets the needs of the patient. Where the data is high quality, so this includes making sure that those samples can go into assays that are generating the needed data to then make those high quality decisions. That if data can't be used to make decisions, no matter how good it is, then it's not particularly helpful to the patient. It's more of a research question. And if the patient is interested in research, that's one thing. But sometimes it's generating noise, at least in Western society, or the US and Europe. What I'm focusing on today in my talk, but but Chris will be going well beyond that, is really what are some of the technology solutions, not people. People are at the center, and you are at the center of finding the right solutions, but there are technologies that can help you. And these are some of the challenges that technologies have been trying to solve. Collecting less, replacing the venipuncture, which can be, a little traumatic for people who either have needle or blood phobia. Collecting by themselves, having more stable samples in order to get those samples, get high quality data out of them. Sometimes you need more precision than what vacuumators can deliver in terms of blood draw, and enabling transport solutions. And there, of course, are some technologies that improve transport, although I won't be touching on those today. Chris may, and certainly I'd be happy to touch on that during the Q&A. So on this slide, I really want to touch on what are some of these solutions and how do they interact with different pieces of what you might be looking for. And I will fix this slide before it's sent out because I realize this Hendriksz is actually blocking the image of this picture. I will try and do it live, but that's not always going to happen. One of the device technologies that has been implemented is that in addition to dried blood spots, there are now some plastic devices, which you can imagine some analytes will be easier to extract off of a plastic than off of a paper, like the traditional dried blood spots, which I'm assuming everybody is familiar with on this call. And also there's some devices that are volumetric. So that includes both this particular plastic device as well as over here on the right. I'm going to make my pointer a little more obvious. Okay. Over here on the right, this also is using the paper substrate that's commonly used in dried blood spot, but uses fluid control through this paper device to ensure that a very precise volume of blood is collected. And the same over here. There are devices that simply remove the vacuum from the tube and are essentially using capillary blood. The SARSET device is one that I'm very familiar with, as are the BD microtiners, which are designed to really collect blood from a smaller volume from a capillary blood lancet. But what I'm showing here for BD is that they also have a fingertip device that connects to their microtainers that essentially makes this whole thing a little bit more integrated, so that you're essentially, for the patient, it's a single device as opposed to a lancet followed by a collection. Here's another paper-based device that you may be able to tell from the subtitled separation on the spot that in addition to collecting a dried blood sample, as shown here, it also separates that blood into different components so that you can then work with something that is a little bit more plasma-like. And then there are devices that are designed to go on the upper arm. I'm showing only one of them here, which is the Tasso device. And you can see that instead of doing a finger or heel lancet, the device itself is put on the upper arm. And then this big red button is pushed to put a lancet straight into the upper arm for collection, which can be less painful for many people than the nerve-rich endings of your fingertips. And I also wanted to include an item which I happen to know is not available in Africa as an illustration of one of the challenges in implementing these devices. This is a device called Tenderfoot, which you may be able to read on the device itself, but it's in relief, so it's hard to it's harder to read than what a lot of device manufacturers do. And what they do is they have different lengths of heel lancets that are designed to be adaptable so that you can pick the right length for your particular infant, generally based on age, but it's by depth of penetration. And instead of a puncture lancet, it is a shallow slice, which in some studies has been shown to be less painful for infants because it's a more controlled distance. And what they try to do is, not really, what they have done is determine the right lengths for different skin depths so that they can do a very shallow cut and get access to the blood capillaries without penetrating as far as the nerve layer. So it is less painful for the infants being sampled. And then there's another one that I wanted to point out that it's not just blood. This is a swab device. It is not a cotton swab. It is a plastic-based swab that fits into a little 96 well sized tube for collecting nasal swabs. They also have swabs for other parts of the body, and then they also have one that is a capillary blood collection as well. And then here is a device that is PIP, stands for pee in pot. So it is a urine collection in a biodegradable collection suit that you can see as the dot in the eye. So there's a lot of different devices, and you can find a list of all of the ones that we have been able to identify through PCSIG on our website. And that includes lancets, blood collection, and other sample collections. There's no one size fits all. And here you see the traditional dried blood spot cards. Most of them are based on Whatman paper, but many companies have rebranded versions and different coatings. And you can see in this slide that there are different coatings, even on the dried blood spot cards, that are available for different purposes. So even dried blood spot is not a one size fits all. So the technologies that are developed are really generally developed to solve a very specific particular problem, which means that if your problem is not the same, your solution may not be the same either. So a really simple, straightforward example is that the dried blood spots are excellent for the heel prick newborn screening. However, they are obviously terrible for doing complete blood counts because all of the cells will be lysed on the paper and not usable to measure what your blood counts were. So what that means is that alternate approaches do solve different problems, and there is no single approach that will solve all problems. So on the previous slide, you may guess that some of those solutions are not inexpensive. They have a heavy cost burden regardless of the geography relative to a vacutainer. However, sometimes the total cost can still be lower if you can decentralize the collection. So the cost of the device might be more on paper, but that doesn't necessarily mean the cost of the full process is. And so all of those factors can determine what solutions are implemented. And we would like to, of course, encourage ASLM to think about some of these questions, both for our Q&A section today, as well as for the panel discussion we are planning on the 7th of May, same time, right here with you, which is, who are the patients that are currently underserved in Africa? You know that far better than I do, coming from the US. I may have greater exposure to some of the technologies, but without exposure to the challenges as a day-to-day capacity, I am not able to advocate for what you need as adaptations of the solutions we have, other than my constant refrain to manufacturers that they should be manufacturing and selling their products in Africa for your ability to use them. Are there solutions that haven't been possible before that patient-centric sampling opportunities can help solve? Do the existing technologies, and which ones, help solve challenges that African scientists yourselves are facing? And which challenges none of the existing solutions work for, but you have an idea in your head of exactly what performance you need from a device, including cost, and can Patient-Centric Sampling Interest Group help you advocate for that with manufacturers or researchers, or even help you develop your own new devices? So one of the reasons that I personally am excited about working with scientists in Africa on this is that there is a huge opportunity in Africa relative to the US, where I am based. In the US, there is a huge barrier to innovation due to maintaining the status quo. In the US, there is extremely heavy capital investment already in vacutainer automation. So these labs, these big clinical labs, have absolutely no motivation to move to alternate technologies that make those automation pathways obsolete. That's where they're getting their competitive advantage in pricing. So they certainly don't want to give it up in favor of a process that is more friendly to the patients, if it means that they have to invest a lot more. So the other aspect is, in the US, assays that are already approved for 510K, which is their approval pathway in the US. If you then change that method to use an alternate sample, then the lab has the burden of conducting additional validation for that assay, which again is different cost. I will quickly go back to the question about why is dry blood spot terrible for CBC? And the reason is because when you put the cells on the paper, to get the cells back off the paper intact, it has been pretty much unfeasible. That the extraction buffers essentially, or even the process of depositing the cells on the paper, causes the cells to lyse. So once they're lysed, you can't count them, because now they're just a mass of membrane, cytoplasm, nucleoplasm, and you can't actually count how many cells were there. You may be able to do other things, and there's certainly alternates that people have been looking into on the technology side that also addresses, can we measure things like the different types of RNA that are expressed in different types of cells to essentially get a different measurement that answers the same question as complete blood cell count. But that's a different topic that I can get into today. But if that's something that people are interested in, certainly something we can touch on in the May panel. So for ASLM, Africa will have different barriers to innovation. And I personally feel, from an outside perspective, that there is a lot more openness and interest in innovation in laboratory medicine in Africa, which is why I'm excited to talk to all of you. That there are cross-country efforts that could potentially be leveraged. The African Medicines Agency has put out some information about cross-country device approvals, and all of these patient-centric samplings are also considered medical devices. How would you build diagnostic access completely differently by putting patients in the center? Is this something that you personally would do something very different? I've seen excellent solutions coming out of these LabCoP, laboratory community of practice webinars that I've attended as a interested party that I would never have thought to build in the US. And so you know already many different ways to innovate that are based on your realities. Who are the stakeholders in diagnostics access and reimbursement? In the US, the stakeholders are, it's a lot of money, and it's private industry and it's government. But that's not necessarily who it is in Africa. And then the regional nuances. This is why I put the outline map of Africa, which I apologize. It is not, the borders are not up to date with all of the existing countries in Africa. However, what I wanted to emphasize is that in the US, while we might have 50 different states, there is an overarching regulatory framework. And in Europe, there is the European Union. And in Africa, well, there are strides to have a cross-continental. There are a lot of regional nuances, even if the governance is regulated. The reality of patients and people are different depending on where you live. So what I wanted to touch on is what are some of the shared principles of access for all patients in all locations? What are some of the potential applications that come to my mind when I look at this from a outside perspective? And these are some of them. Infectious disease is a huge potential application because there are opportunities both in pathogen identification, serological surveillance, as well as treatment monitoring with HIV viral load. If you can test people in where they are rather than in a centralized location, you can reduce the risk of spread during the testing event itself, as well as potentially getting better sample collection close to the potential infectious event. What about some of the diagnosis that is happening too late in the process or not frequently enough to help patients? Would it be helpful to understand risk factors? Are the diagnostic trees incomplete because people who are at risk aren't getting referred as frequently as they can to the specialty labs? Are there opportunities to monitor disease progression in order to know when the treatments need to be changed? And then another element that I think is often overlooked in many places, including the US in pediatrics, is establishing what normal ranges are. And in order that to do that, you need to have samples from this collection. So you would need to be able to collect samples, and are those normal ranges accurate? Are they establishing it for the full range of patients right now if you're only collecting samples from a limited population that happens to be close to the centers establishing these